So in order to understand, what PCR actually is, because PCR is polymerase chain reaction, and it's such an important, I guess, method in terms of developing really good diagnostic assays. So before we talk about PCR, we really got to really understand, the notion of what gene expression is. So gene expression is just a general term that, a lot of the scientists like myself, RNA biologists or molecular biologists use, in the context of describing how a gene is actually just expressed from a segment of DNA into a protein and some of the regulatory functions, that control gene expression. So if you look at this diagram here, you've got the cell, you've got a mammalian cell. So it could be an endothelial cell, it could be an epithelial cell, it could be a cancer cell, it could be any cell. And within that cell, there's obviously DNA. Now, DNA is storage. It contains the information encoded within its double-stranded helix, but it's just a storage product. So in order for DNA to actually be read, there's a process known as transcription. Okay, so what happens in transcription is there are particular sequences upstream of the gene that you want to make. It's known as the TATA region. It forms what's known as the TATA box. Then there's a recruitment of all these proteins within the nucleus of the cell. What happens is that the DNA is bends around this enhancer, who recruits this big protein known as RNA polymerase II. What RNA polymerase II does is that it binds to your DNA, it separates out the two strands and makes a copy of one of the strands, okay? So when it makes that copy, it makes something known as RNA, as opposed to DNA, which is two-stranded. RNA is a single-stranded molecule. That RNA then is transported into the cytoplasm of the cell where it undergoes translation. It gets picked up by another unit here, the ribosome. Okay, the ribosome recognizes this RNA and then it makes protein using 3 nucleotides, and then it attaches amino acid to each of those particular 3 nucleotides. So these proteins come together, everyone. They form molecules. So the proteins come together, form molecules which form structures. The structures then form cells, and then cells form tissue, etc., etc. So this whole process that you see here is something that we describe as gene expression. It's really important to understand because you can actually design diagnostics which pick up mutations in your DNA, or something's, one of the particular nucleotides might be mutated within a certain region. It also measures gene expression. So when a gene is expressed, it's expressed at the RNA level because DNA is static. So designing a diagnostic which measures gene expression at the RNA level, that's what the COVID-19 PCR test does. And also then there's the protein, which is the ELISA test that I mentioned previously for COVID-19. So the next part is, okay, so sit back. I've got a video here. It's a really nice little video. It only runs for a couple of minutes, but it actually shows you it actually describes transcription a lot better than what I did because there's animation behind it. So I'll just hit the play button now.
Is there supposed to be sound to the video, or are we just reading text?
Yeah, guys, you can't hear the sound?
No.
Nope.
Oh, okay. That's weird. I got my sound turned up all the way here.
In Zoom, I think you have to enable the sharing of sound as well.
Yeah, can you guys hear me now?
We can hear you, just maybe not the video.
All right, tell me if you can hear the video.
Yep,
You
that
hear it?
works.
Transcription is the process of making RNA in a genetic code. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. A transcription unit is the Tata box. An enhancer region may also be. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the genetic factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2 near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for binding to RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into mTOR. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors release at transcription points. When the end of the transcription unit is reached, the RNA polymerase dissociates and a newly formed strand of RNA is released.
Okay, can everybody still hear me?
Yes, sir.
Yep, okay, great, thanks. All right, guys, so we talked about, we talked about DNA, we talked about RNA, and we talked about protein. So, this here just looks at, both DNA and RNA. That's something known as nucleic acid diagnostics, and obviously protein, which is macro molecules. So as I've told you previously, your PCR test that you get for COVID is targeted at the RNA because it measures expression of the viral genome, whereas your rapid antigen test is directed against the protein because it measures your own antibodies against a particular virus. Now, PCR allows you to actually measure RNA. We also use PCR to look at DNA as well, but in the context of looking at mutations within the DNA sequence. Yeah. Now, before I talk about PCR in particular, there was a couple of really important events that happened in 1976. One event was the movie Rocky Balboa. Rocky came out starring Sylvester Stallone, so that's a great movie. For those who haven't seen the movie, treat yourself, watch the movie that came out in 1976. Steve Jobs also formed Apple in 1976 as well, but the most important discovery was when scientists were able to find a polymerase and describe it as a Thermo aquaticus. So here is the bacteria. This bacteria lives in hot vents around the world. What scientists were able to do is, for the first time, they were able to isolate a temperature or heat stable DNA polymerase. So this is known as Taq DNA polymerase. And what that meant was you could change the different temperature cycles of a PCR, and then you would still have a stable enzyme or a stable polymerase to actually do the PCR reaction. So we can't use a human polymerase like the ones that we find ourselves, because our polymerases are very temperature sensitive because we're at 37 degrees, but Thermo aquaticus, the polymerase, they can go up to 100 degrees, maybe 105 degrees, but also can go back down to around about 37 degrees as well. So the polymerase itself has a high utility, but it wasn't until 1984. So the polymerase came out in 1976. It wasn't until 1984 that Kary Mullis, he was a scientist. He was a chemist. He was working for CETAS Corporation when he actually commercialized this enzyme, this Taq polymerase, for widespread use in what's known as PCR today. So I just want to quickly talk about the inventor, Kary Mullis, because he's actually, his invention or his idea changed our world. And, you know, the saying one discovery can change worlds is really pertinent when you apply it to Kary Mullis. Now, he won the Nobel Prize in Chemistry in 1993. He was born in just after Christmas at the start of World War II in the US, and sadly, he died in 2019, a couple of years ago. Now, you know, there's a couple of stories. If you do get a chance, you know, do look up Kary Mullis and his story and his life story, because it's a fantastic read. He was an incredible genius. Some say that he was an intolerable genius. He was very hard to work with. Throughout college, you know, when he was a college student, during his memoirs by his friends and himself, he was an LSD proponent. I don't know if anybody knows that's a particular drug. So he quite, you know, he used a lot of LSD as well. And, you know, being a chemist, he also synthesized some LSD during his college years as well. And funny enough, it was his synthesis of LSD which got him a job as a chemist at CITES Corporation. Not the best way to get a job, but I guess that's how it worked during the 80s and also the 70s. Now, the idea that Kary Mullis came up was, you know, he had Taq polymerase there. He was working with the polymerase, and he came up with the idea while he was driving late at night on one of his trips home. So he was thinking, like, you know, we've got these two pieces of DNA here, you know, the double-stranded RNA. We've got the Taq polymerase, which, you know, enables the reproduction of those particular strands. Then he said to himself, what if we design primers? So primers are short sequences which are complementary to sequences of the DNA. And he says, you know, what if we design primers at both ends of the DNA, have the polymerase in the reaction? As a result, those primers then could guide the polymerase to synthesize new products of DNA. So that was his first incredible idea. Then he sat back while he was driving again and thought, gee, what if I could repeat this cycle? So, you know, we've got the first cycle of PCR. What if I did it again? And that was the breakthrough, because PCR is the amplification of DNA or any genomic material through successive cycles. And that was only made possible through the discovery of Taq polymerase, the thermostable enzyme, and also through Kary Mullis' discovery by putting the primers together and cycling that through several temperatures into what's we know as a PCR cycle today. Okay, so just quickly, this is just a historic overview of what's been discovered in terms of methodology. So I might just look at some of the really pertinent discoveries that's happened throughout history. In 1953, there was the discovery of DNA by 3 scientists. So that was a really important discovery of the DNA structure, which then led to our ideas of it being a double-stranded RNA structure. Then led to other ideas such as how to sequence the development of the sequencing techniques by Fred Sanger in 1977. Obviously, Kary Mullis came up with the invention of PCR by identifying primers, the use of Taq polymerase in 83 and 84. In 2001, the human draft genome came out. That was super exciting for us, you know, involved in research. For the very first time, we had a map of the entire human genome. We could understand where genes came from. We could understand where mutations happened in the genome. We could identify regions of the genome which are known as hotspot regions, which are prone to mutations and which give rise to disease. In 2009, you know, sequencing has come a long way on the back of PCR as well. So we had next generation sequencing, or NGS, for detection of human diseases. And in 2011, just over 10 years ago, you know, we've been bringing sequencing to the clinic for diagnostics.